Yeah, even I was missing the practicals a lot because of some
Yes.
unexplainable problems. I was not able to join you all. Um, Anyways, at least now I'm able to meet you all. Never mind, we'll make it up to the loss that we have done. Um, any doubts regarding the other subjects? Also, you can just contact me. For now, we will start with our aquaculture. Um, and I hope you people have your books in front of you all. Applied freshwater aquaculture is a topic that we are talking about.
Yes, ma'am.
Yeah. Can somebody tell me the index or the topics of this subject? Because I have one in my hand, but still, I just want to know if that is the same topics that I'll be covering. So, are you all having the book in front of you all?
Yes, ma'am.
Can you tell me what is there in block one? Uh, what are the topics we have to cover? Block one.
Yes, ma'am. Fish breeding and seed production, freshwater aquaculture production technology.
Yeah.
Yeah, no, in first fresh fish breeding and seed production. Under that, three
Yeah,
units are there.
units are there, right? That is fish breeding, endocrinology, and seed production, right?
Yes, yes, ma'am.
Then the next one we have is extensive, semi, and intensive cultures,
Ye-
prawn culture, pearl culture. Then you have production system management and finfish pathology. Yeah, so that
Yes,
is the
ma'am.
topics that will be talked. Okay, so coming to the first one, I'll be sharing my um PPTs for y'all so that it will be easy for y'all to understand what exactly we are talking about. And in case you people have any doubts in between, you can just stop me and we can continue with the class. I'll just explain and then we'll continue with the class. Okay. So are you able to see the PPT in on your system?
Yes, ma'am.
Right. Okay. So I think you can see even the changed one also. So no problem with that. Yes, Aisha?
Yes, ma'am. Yes.
Right. Okay. So coming to freshwater aquaculture and the production technology over here. Here, what we are going to talk about is why are we talking about freshwater aquaculture? Um, any problem, girls? Because I can see some hear some background noise. No
No,
problem.
ma'am. No.
Ah, okay. Now, coming to aquaculture. So, I'm sure you must have done aquaculture practicals in your practical classes, and you people must have talked about some important indigenous fishes and also exotic fishes. So, when we are talking about indigenous fishes, I'm sure you must have talked about katla katla, labio rohita, and Sirina mrigala. Am I right?
Yes, ma'am.
Right. And then exotic. Can anybody tell me what are the exotic fishes that you all had done? There were three exotic fishes, if I'm not wrong. Forgot. You people had your practicals just recently, right? Tinofaringodon idela, remember? No, did not remember Tinofaringodon idela. Then you have this silver carp, and also you had
Yes,
this
ma'am.
Cyprinus carpio. Remember?
Yes, yes,
You've
ma'am.
plugged into Nirvana. Bluetooth mode.
Okay. If y'all can mute others, if y'all can mute the ones, because that's disturbing my class. Okay, so here, why we are talking about aquaculture is aquaculture can be of three types, that is freshwater aquaculture, brackish water aquaculture, and marine aquaculture. But here in this class, that is your MSc, we will be concentrating mostly on freshwater aquaculture. Why? Because as we all know that we are on the Deccan Plateau, Hyderabad, on the Deccan Plateau. So even though if you're on the Deccan Plateau, you can still have this aquaculture, that is freshwater aquaculture.
Powering off.
If you're going to go to the marine ones, there you will have to again stay in some ocean places where you have to do this culture over there. But here within Hyderabad, if you can do, or within the Telangana state, if you want to culture, yes, you have this freshwater aquaculture. Here we'll be talking about how you can do the production technology. Technology is how science is going to be used for increasing the production of the freshwater cultivation, that is we'll be talking about. So here, if you can see, as the image over here, you can see, so you can have a small place just behind the backyard, or even if you have a small land in your villages, so you can do this pond culture. If you have a big areas that is in the wild, you can do this culture. And also sometimes you can also use this integrated aquaculture. So what do you mean by integrated aquaculture? So here, when we are talking about aquaculture, we are not just talking about fishes. We are also talking about the prawns. We are also talking about the shells. We are also talking about a different type algae. So name anything that is going to grow in the water, that is aquaculture. Aqua means water. Farming in the water is called as aquaculture. So here in this integrated one, you can not only grow fishes in them. Along with the fishes, you can also grow your the crabs. You can also grow your prawns, the different types of prawns. It can be shrimps, it can be prawns, it can be even lobsters. It can also be shells for pearl culture. You can do the same thing in this integrated aquaculture. So here, particularly, today's topic will be talking about fishes over here, and why India is giving so much of importance. We'll be talking about that in this the next slide over here. So as you can see, India's annual fish production has reached all-time high, that is at 19.77 million tons. When I was talking about this topic just 2 years back, that is in 2023, I was talking about this at 11 million tons over there. Within 3 years, you have this 19.77 million tons. So you can imagine what is the importance of this fish production in India, and why we are growing them, or why India, the government is giving so much of importance to this aquaculture. You must have heard this Pradhan Mantri Matsya Yojana. Then also you must have heard of this Blue Revolution, where government is giving a lot of importance to the production of fishes over here. And because of that, now you can see India has become the second largest aquaculture production among the top 10 producing countries in the world. So you can see that much of production that is happening in India. And also the government is also giving so much of importance. You can also always have this as a small scale industry. If at all you have some places, you can also think of growing them by yourself, and you can also become owners of aquaculture. Because if you can see in this, the sectors experienced a massive 1.6% increase in the production. So imagine the increase in the production, that is at 1.6%. 100% may 1.6, definitely it's a massive one. Why? Because India is turning into fish eating population. Now, why fish eating population? Why? Because fish has highest amount of protein. The percentage of protein is highest in the fishes when compared to the chicken or the mutton over there. And the most important point is over here, fishes has less fat. That is, even though they have fat, they have this beautiful fat called omega fatty acid. If you all must have heard of omega fatty acids, which is good for our body over there. So Indian population is slowly moving from red meat to fish eating population. So you can just cash in on this type of production over here, and hence you can also have a small scale industry. So your education, your topics that we are giving, the university is giving you all, is going to be useful for you all for the future. It is not just the topic, that another type of a topic that you study and you leave it. You can take it up as a small scale industry, grow up on your own, have your own industry, and you can become owners. And yeah, you can also become multimillionaires over there. Okay, coming to the next topic over here. Yeah, nearly 70 to 75% of this production is from freshwater fish farming. So you can imagine what is the percentage of freshwater fish farming is all about. So as I told you all, among these important fishes, we were talking about the Indian indigenous fishes, that is katla katla, Labio rohita, and Sirena mrigala. And among the exotic ones, you can talk about Tinopharyngodon idela. Then you also have Hypothalmicus mollitrix. Then you have also Cyprinus carpio, these three. So these six fishes can give you all a maximum amount Along with this, you can also talk about the lobsters that you had talked about. Two lobsters you were talking about in your class, Macrobrachium rosenbergi and Macrobrachium malcolmsoni, if I'm not wrong. Yeah. So these two lobsters we were talking about in integrated fish farmings over there. So here, when you're talking about freshwater, when I'm talking about lobster, I'm talking about not the marine ones, but freshwater fish farming. So under freshwater, you have these two important giant lobsters, that is Macrobrachium rosenbergi and malcolmsoni. So these two can be grown in freshwater fish farming. Now, if you're having a hectare, so if you're having a land which is having a hectare, so what is the amount that you can produce per hectare per year? You can grow about 4.77 tons. You can imagine what a ton is. So you can grow 4.77 tons per hectare. So if you have a 2 hectare, then imagine you can become definitely a millionaire or multimillionaire very soon. That is provided you have an interest in aquaculture, and also you should learn the nuances of how you're going to take care of these fishes. And this is what this aquaculture topic is going to give you all an insight into how you can start your small scale industries, if at all you all are interested. Okay, so here we are. This is what I was talking about, the major carps, the katla, rohu, and mrigal. And how many types of freshwater fishes India has? Although you have thousands of them, mostly 838 freshwater fish species occurs in India. And among that, 450 fish are categorized under small indigenous species, that is, which can grow between 25 to 30 centimeters. 30 centimeters, almost the large scale, the big scale, that size that you have. So 450 are of that size, and the rest of one a little bit bigger in size over there. And who are the top leading states of India? Of course, Andhra Pradesh, then comes West Bengal, Uttar Pradesh, and Odisha. Then when you're talking about Andhra Pradesh is famous for shrimp. If you remember what a shrimp is, the shrimp is a smaller size of the prawns. And what are prawns? Prawns are a little bit smaller than the lobsters. I'll show you all the difference over here. So that's a shrimp over there. A shrimp is a small prawns that you find over there with smaller legs over there. They have almost 2 pairs of claws. The prawns have 3 pairs of claws. If at all you must have cleaned it up, you must have seen. You will have the legs with claws over there. And yes, the lobsters, that's giant lobsters that you have over there. And this is the large claws, the second pair of claw, which is going to be very large in the lobsters. And that's how you'll be able to hold them with the help of those claws over there. Okay, so highlights of this industry, because if you're going to talk about small scale industry, you should also be knowing what is the amount that is being given or present over here, and what is the amount you're going to earn in the future. So here at the fishery sectors, roughly 7.43% of India's agriculture gross value added is contributing, and this is also going to give livelihood for almost 28 million people of India. So imagine 28 million people depend on this fishery sector, whether it can be freshwater, marine water, or also it can be brackish water. So 3 types of culture. So 28 million people survive only on this fishery sector over there. And export. So when you're growing so much of fishes, what are we doing with so much of fishes? It has to be exported, definitely, right? Because we are not going to eat the whole thing. India is not going to eat the whole thing over here. So what is the amount of export that you're going to do? 350 seafood products are being exported to how many countries? 130 countries. So 130 countries, India is exporting its seafood over there. So imagine the type of business India is doing right now over here. And what is the earning? It has scaled up to 64,400 crores, or close to 7.4 billion US dollars annually. So we are talking about billions over here. So mostly over here, which is the one that is going to be exported? Frozen shrimp being the primary export commodity in this aquaculture. Export. Import, yeah, in India we can also talk about the other fishes that we're talking about. So this is a difference between a shrimp, a prawn, and a lobster, for you all to understand. When I'm talking about shrimp, don't get confused with a prawn and a shrimp. is a little bit different in the size. And of course, as the lobster, the big lobsters, the exotic ones over there, if you're going to grow this lobsters in your aquaculture, then definitely you people are going to earn in billions. Why? Because one lobster of this size, the big size, almost say 25 centimeters to 30 centimeters, also you can get the big sizes over there. So one lobsters in 5-star hotel will cost you easily around 2,000 or 3,000 per piece over there. So imagine if you're going to grow lobsters, you're definitely going to hit the bullseye in this culture. Yeah. So when I'm talking about the seafoods not only includes the fishes over here or a prawns over here. You can also see the crabs, the shells, the different types of sharks that you have over there. So these are the different types. I must have seen also this over there. If somebody can name what exactly is this that I'm talking about, you can just get back to me. If you must have seen this even in the fast food, the malls and all, they sell this also over there. I want one of y'all to name what exactly that one is. As I'm talking, just get back to me when what exactly is that. Okay, coming to fish breeding and seed production. So now that you have understood what are the topics that you're going to talk about in the aquaculture, how you're going to breed them and how you're going to get the seed production. So here, when I'm talking about seed production, the seed production is not, I'm not talking about the seeds of the plant, but I'm talking about the eggs of the fish. Because when you're going to do an aquaculture, you need some raw material. Like for example, the farmer needs a seed. He has to sow the seed, he has to water them, and then he grows those plants over there. The same way in aquaculture, you need the eggs over there. So if you have the eggs, you can start your aquaculture. And also you need a place where your big fishes can breed, so that you have a constant supply of eggs for the next harvesting or the next cultivation to begin after the harvesting of the big fishes over there. So what are these? So in an aquarium or a pond, so this is a pond, say, or an aquarium like your the small aquariums that you might be having in your homes, whoever is interested in growing those small fishes over there. Successful breeding largely depends on nutrition and environmental condition that are sometimes very specific. Nutrition is what type of food you are going to give these fishes to grow, and what is the type of environment condition that you're going to take care of. So in your practicals, if I'm not wrong, you must have done 4 or 5 experiments over there. You must have done oxygen estimation, then you must have done the calcium, the magnesium estimations, the alkalinity of the water, the salinity of the water. All these you must have done in the practicals. So that is the environmental condition that we are talking about in this particular topic over here. So if the oxygen is depleting, then that means then the fishes are going to die. If the salinity is increasing, again osmosis can come into effect. And if at all the alkalinity is increasing, then there is a different kind of a problem over there. So you have to constantly keep checking time and again, that is every 2 to 3 months, you have to keep checking the water condition, so that the water is having enough oxygen, and it is also maintaining the salinity and alkalinity in the water, along with the minerals. So your minerals, what are the important minerals that you must have checked in the practicals? You must have checked calcium, you must have to also checked magnesium over there. So these two are again important. Why calcium? Can anyone answer me this question? Yes. Why did we check the calcium in the water? Did we do this experiment? Calcium estimation of calcium. Anybody in the class?
It increases the hardness of water.
Increases the hardness of water, and anything more? Anything more? Why is calcium important?
Calcium is a source of vitamin D.
Vitamin D. So it helps in shell formation of the
Shell.
eggs. Yes. If the shells are not good, then definitely you will not be having enough seed production, right? So all these have to be maintained in the water. Very good. And when you're talking about in the wild, so that's in the wild means you're talking about the river beds or the rivers where you can directly go fish them over there. So this is the places where you can also grow them like that, having the cages over there in the wild. So these are the cages where you can just keep all your fishes in one place, doesn't allow them to move away. Now, why do we have to grow grow them in the wild like this with the cages is because you want to give that natural environment for the fishes to grow so that they can breed naturally. If you had done this practicals, you must have also seen that we also did one particular experiment where you are going to induce the fishes to breed. So if you remember, there was one topic, induced.
Disconnected.
Yeah, so in the induced breeding, what we were talking about is we take out the pituitary gland of the fishes, and then we induce this hormone into the captured fishes. So these are the captured fishes over here. So you induce them as so that they'll help in breeding. But here in this particular wild, you don't have to do that because the water current, the temperature, the atmosphere, all those are conducive for the fishes to breed naturally. So this is one more important specific point where you can grow them in the wild. The amount of daylight, the availability of the specific food sources can also have a major effect on the development of the fishes over there. And of course, during rainy season, the water may wash extra nutrients, that is from the mountains or nearby places. Extra nutrients can come from different parts of the places over there, and that will also increase the abundance of the food that is available, which can be ideal for breeding conditions of the fish. Just give me a point, girls. I have to add this charger for my cell computer. Just give me a sec. I used a lot of my energy, so sorry for that delay. Okay. Now coming to the method of reproduction over here. So you're again, when you want the seed production, then definitely you'll have to talk a little bit about how the reproduction occurs in the fishes over there. So most of the fishes, they lay small eggs, fertilized, scattered outside the body. So something like this you must have seen in the water beds, or if at all you had aquarium, you could have seen those eggs being laid or scattered around. The mortality of the young, especially the eggs, is very high. That is, you must have seen when we are talking about katla and labio rohita, the amount of eggs that are laid by a kg fish. If you had read those points, you must have heard it is between 2 to 3 lakhs per female per kg eggs. So all this, there's so many eggs that are being laid, and these eggs are laid just like that in the water, which is easy for the predators to come and eat away. So among that lakhs, only few of them grow to maturity level. So that is the reason why in aqua environment, all those that lay eggs, it is always between thousands to lakhs, depending on the type of the species that we are talking about over there. So in that lakhs and thousands, it's only few of them which grow into maturity. So that is how the nature is all about, because the other fishes also need food, especially the carnivores. So they need eggs for themselves to eat, and along with that, this fishes also have to survive. So only few of them can mature. So imagine if this lakhs and thousands of eggs grow all together, then our water is filled with only fishes over there. So nature balances each and every species in its own ways. So here mortality is also same over here. The eggs are eaten away by other fishes, so only few of them mature. The male fishes, they produce a milk, that is a milky white substance called milk, which is nothing but the sperms. And in females, the eggs are formed in the two ovaries. If you must have cut open the fish over there, you must have seen those ovaries, a pair of ovaries in your practicals when you had done this GSI. The gonadosomatic index and all. So you must have done all those in the practical classes, I'm sure. So those are the two ovaries, and the fishes, the eggs are fertilized internally or externally. Also, two types of fertilization. So what are they? The types of reproduction, here you have sexual reproduction and asexual reproduction. So in the sexual reproduction, definitely you need an ova and a sperm, the process of fertilization in most of the animals. The female lays the eggs over there, or the ova, and the male fishes, they let out the milk or the sperm on the ova over here. So if you can see the eggs over there, and those white color ones are the sperms, which goes and fertilizes the egg. And this fertilization is external fertilization over there. Now, when you're talking about the types of reproduction, the types of reproduction over here, the first one, which we had already talked about, the sexual reproduction. Most of the fishes are of this type of sexual reproduction. And in hermaphrodite, that is hermaphrodite, if you have understood the word hermaphrodite, that is both male and the female organs are present in the same animal. And both of them, sometimes when one female, the female and the males over here, so if one is giving eggs, the other one is giving the sperms over there. So but here in hermaphrodites, both the reproductive systems are present in the same animal, that is called as hermaphrodite. And examples over here are anguilliformes, eels, that is the eels that elongated ones, the eels over there, and the killifishes are the examples over here. Now, here, what is the thing that you have to understand why we are talking about hermaphrodite in the fishes is, it can be of two types, that is protandrous and protogynous. Androus, androgen, male. Gynous, gynecology or gynec is the females over here. So protandrous is, pro is first. That is when a fish is born, when from the eggs, when they are born, they are first males, and later on in their life they become females. Later on in their life, that same male becomes females. So that is protandrous. And what is protogynous? When it is born, the fish is first a female, and then later on it becomes male. So the example over here, what I want you all to see over here is this particular examples over here. The protogynic, that is when the fish is born, first it is a female, then later on in its life it becomes male. In protandry over here, this is a sea anemone. If you remember this, there was a movie also on this sea anemone movie over here. Now this is a male first, and then later on in its life it becomes female. Now there's a beautiful story in this sea anemone over here. Why? Because in a group of the school of fishes, the group of fishes is called as a school of fishes. In that school of fishes, you have only one leader, and that leader is a female. The largest, the biggest size fish is a female, and this female has only one partner, that is a male. The rest of the fishes are all males. Why? Because all the small ones are males, and all these males do not mate with this female. It's only one partner, so that's only one partner which will mate with this female over there. Now, because of some circumstances, if this female dies, the male that was a partner for this female changes itself, grows in size, changes itself into a female, and then that becomes the leader of that school of fishes, and it has another partner. So only one partner. So that is the beauty of this particular protandry and protogyny. And there is one more example of it, that is bidirectional. Whenever they want, they can become a female. Whenever they want, they can become a male. So that is the advantage in these fishes over there. That's the hermaphrodite over here. And parthenogenesis. You must have heard of parthenogenesis in arthropods. What's an example of parthenogenesis in arthropod? Very famous example. If anybody can answer me this. Parthenogenetic. That is reproduction without mating. One example in arthropod. I'm giving you an example also. Arthropod, parthenogenesis, very famous example.
Ants.
Yes, ants and honeybee. I was expecting honeybee. Honeybee is most famous example of the parthenogenesis. Yeah. So what is happening over here? The development into a young ones without fertilization, that is called as parthenogenesis or asexual. So the example over here is a female shark can undergo this parthenogenesis, that is a form of asexual reproduction where the embryo develops from an unfertilized egg. And this is seen mostly in zebra sharks, hammerhead sharks, white-spotted bamboo sharks, that is virgin birth is seen in this parthenogenesis. genesis. So that is the type of reproduction over here. Now, there are different types of reproduction strategies over here. So what are the strategies over there? So the fishes can be of three types: non-guarders, guarders, and bearers. Now, this non-guarders are, they lay the eggs wherever it is pleasing for them. They just leave them over there. And they, if all the eggs survive, then fine. Otherwise, some are eaten away by the predators, as we were talking about. So these are non-guarders, that is, they can be laid over anywhere in the open substrate spots. And there are another type, brood hiders under non-guarders. Brood hiders is, if you can see, they slowly attach their egg on some to the leaves or to the shells that are present inside, or onto the stones. They cover them, the eggs, into these hiding places over there. That is called as brood hiders over there. So this is one type of strategy over there. Then you have the next one, the guarders. That is, after laying the eggs, they guard their eggs. So that is guarders over there. So mostly both male and the female, or sometimes it's only a male or a female which guards the egg after laying. Then this are of two types: substrate choosers. So they choose a place over there. Now, this is a place over there. The female comes and lays its eggs over there. The male spreads its milk on to the eggs over there. And after sometimes, what happens is, after laying the eggs, the female moves away. It is done its with its work. It has laid its egg. It will move away. It is the male, the smaller one, which takes care of the eggs until they hatch. So that is one beauty in fishes, where the male will take care of the young ones. Then the next one is nest spawners, something like the birds which build their nest. The fishes also can build their nest. A beautiful example, if you all know, there is one beautiful fish which does this nest. That is this. Have you all ever seen this shape in the environment? Of course, you can't see it over here. It is in the ocean beds. You find this particular chakra-like structures that is present over here. Now, this type of a nest is built by none other than this particular beautiful fish called as puffer fish. I hope I understand you must have heard of this puffer fish. Why puffer? Because when they puff up, they become bloated up like that. They inflate themselves so that they bloat up like that. Why they bloat up like that is to make the other fishes to run away. That is how their defense mechanism is. They just puff up, and these puffer fish are very toxic again, although it is not going to lead to mortality, but definitely when they bite us, it's going to have a stinging effect. It gives pain over here. Now, what this does is, this beautiful engineering is done by this male puffer fish. It's not done by the females. It is done by the male, just to attract the female. They built the nest to attract the females over here, the puffer fish over there. Now, this beautiful engineering work was a real it was a hidden secret. The scientists didn't know who was the one that is going to build this type of a beautiful structure on the ocean bed. And then later on, they came to know that this tiny fish is the one that is going to build it. Now, why this beautiful shape is, in the center, you have this place, the center. Now, this is a place where the female is going to lay its egg. Why? Because of this type of the shape, whatever the water current is going to be, whatever the force it is going to be, the eggs are not going to be disturbed, because it has a wall around them. So this is the wall around them. All the eggs are going to be staying safe over there. So this is a beauty of the nest spawners, where they nest breeders, where the eggs are being kept over there. Now, when a female sees, and if it likes this nest, then they will come and mate with the males over there. The female will just lay its eggs over here in the center, and the male is the one that is going to take care of the eggs until they hatch. So that is again the nest spawners is the examples over there under guarders over there. Now, the bearers over here, the bearers are, they bear the eggs within themselves. That is called as bearers over there. Till now, we were talking about where the eggs were released outside. Here we are going to talk about those which are going to keep the eggs inside them. That is bearers, external bearers and internal bearers. The example of the external bearers over here, what happens is, the eggs can sometimes be attached to the skin. That is why I'm talking about external bearers. That is, the eggs are not being released into the environment, but they're going to be stuck up to the bodies of the fishes itself. So where all it can be seen? It can be seen as attachment to the skin. It can be seen present in the pouch. It can be open pouch or a closed pouch. The pouch, as in we are talking about not the pouch that you have the purses, but the pouch. For example, kangaroos have pouches. Yes, in the same way, the seahorses also has a pouch over there. What happens is the female seahorse lays the eggs into the pouch of the male. So here the males only have the pouches. So the females lays the eggs into the male pouches. The male later on takes care until the eggs are hatched over there. So again over here, male parenting can be seen over here. And again, sometimes you can also see the eggs on the forehead. The fish carries the eggs on its forehead, taking care of them, because they fear if any big predators can eat them away. So these are the external bearers. Now there is also external bearers of the mouth over here, some mouth bearing also. Now you can see there is a small fish in the mouth of the animal. This is not eating, but the parent is only taking care of the fish inside its mouth. You can also see the eggs over here. The male parent is having the eggs over here, so you can also see this over here. Here again, mostly males, very few females carry the eggs in the mouth. And when they carry eggs in the mouth, you will be wondering, won't they eat them away? Yes, they never eat them. That is, mostly the males have this into their mouth, and the males are not going to eat any food until the eggs are going to hatch. So they are something like they go into fasting period where they don't eat food at all during those period. So that is the importance and the beauty of the fishes of there. Imagine the Pisces, such tiny brain, and they have so much of parental care. So nature is beautiful, right? They teach us so much of parenting. Of course, we humans might have learned how to take care of the young ones from these beautiful tiny creatures over here. So this is external bearers where they keep the eggs in the mouth until they hatch. They are not going to eat, so they are going to fast 3 days, 4 days, 5 days, depending on the species over there. Okay, coming to live bearers. So here we were talking about live bearers over here. So in this live bearers, we're going to talk about internal bearers. Till now we were talking about external bearers. So these are the external bearers over here. Pouch, the forehead, the mouth bearers over there. Now coming to internal bearers. Internal bearers is you're going to take care of them within. So something like your mammals, where we grow the young ones within the womb itself. So in the same way, internal bearers, you have three major categories: oviparous, ovoviviparous, and viviparous type of fishes. So under oviparous, fertilization is internal. It's not external, where the female lays the eggs and the male throws its milk on the eggs over there. That is external. Here, fertilization is internal. Incubation is external. After the fertilization, the eggs are released outside. Incubation is external. That is, incubation is something like the chicken which sits on the eggs. That is called as incubation. Incubation is external. No nourishment from the parents. Examples are sharks and skates. Now you'll be wondering, why no nourishment? Then definitely the young ones are going to die. There's something called yolk that is present in the egg, and this yolk forms the food for the young ones to take it up and grow. So that is, incubation is external. Then the next one is ovoviviparous. Fertilization is internal. Incubation is also internal without nourishment. Again over here, there is no placenta. Something like the humans have the placenta and the umbilical cord where the mother is giving nutrition. So here in ovoviviparous, it is in the egg, but when it is released, it is a young one. That is why you call it as viviparous, ovoviviparous. So how is the development occurring? Again, yolk production. So the egg has enough yolk for the development of the young ones to occur. So that is why fertilization internal, incubation also internal without any nutrient supply from the females. Example over here, whale sharks, skates, and living fossil fish, latimeria. You must have seen this latimeria in our lab. We have the specimen over there in the lab. I suppose you must have seen this also. Then the last one is viviparous, giving birth to young one as such. So here it is fertilization internal, incubation is also internal, nutrient supply is also from the mother. That is, the placental connection is present over here. And examples over here are bull shark and Poecilidae. So this is a small Poecilidae. You must have grown this Poecilidae if at all you people have aquarium, anyone in the group having aquarium, then definitely you must have seen the small fishes over there, which gives that birth to young ones directly. So that is the example of viviparous. So under livebearers, three types: oviparous, ovoviviparous, and viviparous. Now coming to fish breeding and genetics. Now this is a beautiful part that fascinates me. Genetics is always fascinating subject over here. Breeding and genetics. Now why we are going to talk about genetics over here is because when I was talking about technology, this is what technology is over here, where you're going to use science in growing these fishes. That is, if they're going to grow in naturally, you're going to take some time. Say if a fish has to grow 1 kg, you will have to wait for 1 year or 2 years for the 1 kg to grow. So that means every harvest you have to wait for 1 year or 2 years. But science of here says, why wait for so long when you can manipulate its genetics? That is technology over here. So for understanding that technology, first you have to understand what are they made up of, what is the number of chromosomes that they are made up of over here. So here when you're talking about zebrafish, now this is a zebrafish which is a very small one, which can be grown in aquarium also. Although it is very small, what is the chromosome size? The number 50. What is the number of chromosome humans have? Anybody?
46.
46. That is 23 pairs. Each
23
coming from one parent over here. 46. Now 46 to this height. Now this is such a tiny fish, this much only, but it is having 50 chromosome. So size doesn't matter over here. So it can be more, it can be less. Size is not at all a matter. When you're talking about a goldfish, that small goldfish which we have in that small pots in our glass pots in our homes, what is the chromosome number? Can you see that? 100, 2n. What is 2n over here? 2n is diploid number of chromosome. 1n from each parent. One from mother, one from father, 2n. That is 100. So this over here is 100 for goldfish. And goldfish over here are tetraploid. So what do you mean by tetraploid? It is 4 sets. That is 2n from mother, 2n from father. That is tetraploid. That is 4 sets over here for goldfish over here. And you also have Atlantic salmon over here. The salmons are very, very famous in the foreign countries because it's having a lot of nutrient values over here in the salmon. What is the chromosome number? 58 to 60 over there. And in rainbow trout, why rainbow trout? Because the color is almost like a rainbow over there. At 58 to 64, and guppies at 46. So on and so forth. These are some examples I wanted to give you all. These guppies are also grown in the aquarium with beautiful color, different colors you have here. And why zebra fish and technology over here, genetics over here? Because zebra fish is a fascinating fish for scientists because 80% of our genetics is same as that of humans. So zebra fish and we are almost 80% close to each other at chromosome number 50 there. So scientists do a lot of research on this zebra fish over there. So that is the importance over there in the zebra fish. Now coming to polyploidy, something like this. So what is this? Tetraploidy, hexaploidy, pentaploidy. You're going to talk about polyploidy. You must be wondering, why is ma'am talking about plants and fruits over here when you're talking about fishes? I want you all to understand what is polyploidy, many. From 2n. 2n is humans. So if you can see over here, 2x or 1x and 1y. That is 1n from mother, 1n from father, 2n. 1n from mother, 1n from father, xy is again 2n over here. That is diploid. So we are all diploid. If anything is deviating from diploid, if it is a triploid, tetraploid, pentaploid, hexaploid, then that is called as polyploidy. So here, why I'm giving this example over here is we are manipulating the genetics of the fruits also. That is why we are getting this genetically modified fruits over here. So you must have seen in earlier days when you were young small ones, when you had this watermelon, every bite you used to have so many seeds coming out. You have to keep spitting so many seeds over there. But nowadays, just cut it, eat it without any seeds over there. Seedless watermelon. How did they do it? They got this by changing its genetics over here. So they made this watermelon into triploid over there. So triple X over there. So triple X will not have seeds. Same thing with banana. Earlier, if you would have eaten a banana, in the center, you must have seen some seeds over there. But nowadays, no seeds at all. Seedless banana. Now, when you're talking about this blackberries, very small ones. Now you have bigger ones. Peanuts, also big in size. And also the strawberries earlier, if you remember, you had only the small ones. Nowadays they're selling on the streets outside over there with that big size. How? Again, genetics over there, we have played with its genes over there. Sweet potato, increasing its sweetness, increasing its size. Hexaploid, so how many hexaploid x's? And in strawberries, octaploid. So polyploidy means we have played or meddled with its gene to give us either a big size or seedless, or nutritional value has been increased. The same way we have also done with the fishes over here. So this is what I'm talking about over here. Now you will understand why polyploidy in aquaculture. So here, what we did was, or what a scientist did was, they took two fishes over there. One, they named different names over there. You don't have to worry about the names over here. So this is a 3N AUT, and that is an RCC. This 3N AUT is the number 3N at 150. So that is triploidy at 150 number of chromosomes, and RCC is diploid with 100. Now this is a goldfish. Okay, so and the same goldfish was changed to triploid, becoming a white color. So the gold is no longer gold color, but it has become a white color. Now, when they mated these two together, if the genes were mostly of RCC, you get a diploid. So this is a diploid, you get a diploid over there. If you get a 3N over here, the triploid over there. But if you get a combination of these two, now you get a tetraploid at 4N. This is 3N, this is 2N, but neither of these two are of the character that you find in this offspring. And what you got is a tetraploid 4N at 200 chromosomes, right? So it got 200 chromosome over here, the 100 from here and 100 from here, you got a 200 chromosomes of this particular type of a fish. Now you'll be wondering, why did we play with its genetics? Why did we play with genetics? You wanted to achieve sterility. You wanted to achieve accelerated growth. That is, you want the fishes to grow faster. Why wait till 1 year? If you can grow them within 6 months and harvest them and get the money faster, that is accelerating growth. Improve flesh quality. Flesh quality is you get more amount of flesh, more amount of protein, less amount of thorns also. Sterility. Why sterility? Because you don't want them to have the fertilization between two different, and again changing its genetic potential over there. So that is why sterile ones over there. The same way in humans also, if you remember when we are talking about the chromosome, XXXY, XXY, YYX, XYY, all these are sterile humans. Okay, so same thing. If you're going to meddle with the genetics, then definitely there is some drawbacks and also advantages in them. Okay. Now coming to hybridization in aquaculture, why? Again, as I told you all, faster growth rate, higher disease resistance, better temperature tolerance, because the temperature also plays a lot of role over here, because fishes, if you have too much of temperature, then definitely the fishes are going to die. So you have to take a better care of them. And that is the reason why if you're going to meddle with this genetics, they can also bear up with the temperature. So the best fishes that can bear up with all temperature in natural is Cyprinus carpio. That's a European carp. Cyprinus carpio is such a type of a fish. That's why it's called as common carp. It can grow anywhere in this world. You grow them in a oxygen less water, it will grow. You grow them in a crowded place, it will grow. You grow them with high temperature, low temperature, salinity, difference in the salinity, they will grow. That's why it is called as common carp, or it doesn't care at all about its temperature or whatever it is. So that is one particular example of the fish which can grow wherever you want to grow them. But here, when you're talking about katla, labia, rohita, they need a lot of importance to be given in the nutrition, the place where you're growing them. So here, when a hybridization occurs, accelerated growth, that is, they grow much faster than their parent species, reducing the time required to reach the market, as I told you all. Controlled reproduction, crossbreeding, certain species can produce all male population. Now, why all male population? What is the fun in having all male population? when you're going to grow only all male populations, these fishes will tend to grow only in size. Males, the anatomy is as such, they grow in size, they grow faster. If it is only females, then the females have to divide its energy for reproductive system and also itself. So there the partition is occurring and the development of the fish slows down. So if you want only for market value, then this is the best way of producing all male population. Then hardness. Hybrids frequently display stronger immunity. So this is also very important, because when you're growing fishes, there is one more important point. It is not just hunky-dory. Okay, you bought the seeds, you grew them, and then you're going to harvest them. It is not so. If you're not going to take care of them, then definitely there are different types of diseases that affect the fishes also. So you have to be very careful in checking them time and again. So here, this hybrid ones, you can grow a stronger immunity fishes also, which will have tolerance to diseases and tolerance to stressful environment. Quality of the flesh is also one of the important one. If the quality of the flesh is waste, then nobody is going to buy them. The better flesh quality, the better yield and better advantage on those fishes. The examples. Now, what are some examples, just two examples I'll give you under this hybridization technique over here. The first one is a catfish. You must have seen this catfish on the roadside where they have the small plastic pond-like structures, the small ones over there, and they have this catfishes present in them, that elongated ones, ribbon-like elongated ones, and you must have also seen this whiskers on them. So all those fishes which are having those whiskers, they are called as catfish. So the example over here is a cross between the channel catfish and a blue catfish gave a fast hard ones. Hard one is immunity, better immunity, fast going fish with excellent fish quality. Now this is the hybrid catfish. Now you can see the difference over here, the hybrid catfish. As I told you all, catfishes are all elongated like this. But here in this hybrid one, you can see they are oblong also. That is, the amount of flesh is also increased over here. That is why the hybrid catfish over here. Then the next one is hybrid tilapia. Tilapia, you must have seen even tilapia in our college. You had this specimen also. How you differentiate tilapia? The dorsal fin and the ventral fin, they are ribbon-like structures. The fish resembles like a bow-shaped ribbons, right? So that is a tilapia over here. Mating a Nile tilapia with a blue tilapia, what you get mostly male offsprings. Again, why males over here? Males, why? Because you have a faster growth rate in them. So tilapia, between the Nile tilapia and the blue tilapia, will give you a male offspring. Predominantly, it's only males. So this is again a hybrid tilapia. So that's examples of hybridization techniques over there. So this was some of the important informations that we have for the aquaculture production. Now coming to endocrine. Till now, any doubts, students? You have any doubts over here, or if you don't have any doubts, we'll continue with the next topic over here. Yes, shall I continue the next topic?
Yes, ma'am.
Yeah, coming to the endocrinology of reproduction and breeding of the common carp. So here we are talking about the breeding of common carp and endocrinology. So whenever there's endocrinology, then definitely we are going to talk a lot about hormones, endocrinology and hormones. So here, because we are going to do a lot of breeding, lot of hybridization techniques and all those, you have to understand a little bit of the brain of the fish over there. So that is the reason why you have this structure of the brain over here with the optic olfactory bulb over there, the cerebellum, the optic lobes for the eyes, the pituitary glands over there, and then the hypothalamus that is present over there, the cerebellum over there. So this is the place where you have the brain of the fish. You must have, I suppose you must have done this dissection of extracting the brain of the fish in your practical class over there. Now coming to exact picture of what exactly I want you all to concentrate on in this fish brain is, I want you all to concentrate on this pituitary gland, the hypothalamus and the thalamus part. Now this is the place, the forebrain, where you have this the thalamus, the hypothalamus and the pituitary gland. Now, these are the ones that is most important for endocrinology point of view. The others are also important, but here, when I'm talking about endocrinology, I'm interested only in this part of the brain, fish brain, that is the thalamus, the hypothalamus over there, and the pituitary. The thalamus gives information to the hypothalamus gives information to the pituitary, and the pituitary gives signals to the thyroid glands. So what is that all about? So here, when you're talking about the hypothalamic pituitary gonadal axis, so here, hypothalamus, pituitary, and gonadal axis. What is gonadal axis? Giving information to the gonads. What is gonads? Reproductive organs. So who's the one that is going to give us information? It's the brain that is going to give information to the reproductive organs, whether to mature or whether to stay immature. So that is what we are going to talk about over here. A little bit of this topic. Of course, it's going to be very interesting and information. Now, the hypothalamus controls the synthesis and the releases of the hormones. Why hormones? We are talking about endocrinology. Hormones is the one that is going to give, say, for example, growth hormones, thyroid glands, growth hormones, or whether the reproductive organs development, or it can also be your insulin development. So different hormones. Here, we are going to talk about the gonadal axis, hypothalamus, pituitary, and the gonads. I'm not talking about the other hormones over here. So what are they? So these are the chemical messengers resulting from the central nervous system. There is a brain over there. Gonadotrophin releasing hormones. GRH is the gonadotrophin releasing hormone, which is released from the hypothalamus. So you have this hypothalamus over here. So the hypothalamus is going to release the GRH, that is a gonadotrophin releasing hormones from the hypothalamus, stimulates the release of gonadotrophin from the pituitary. So here, hypothalamus is giving information to the pituitary. Now, this pituitary is getting the information from the hypothalamus to release that GRH. Now, this GRH goes and gives information to whom? To the thyroid glands over there. So this is the process that goes on over there. So it goes and gives information to the pituitary, and now this goes and gives information to the thyroid glands, which is in turn going to give rise to thyroid stimulating hormones. How? Pituitary hormones includes adenocorticotrophin, prolactin, growth hormone, thyroid stimulating hormones, and the gonadotrophin, gonado for the gonads of the GTH. So the two forms of gonadotrophins isolated from the fish are GTH1 and GTH2. What are they? Gonadotrophin 1 and 2. And these are the analogous to the mammals also. What are they? The mammalian follicle stimulating hormones and the luteinizing hormones. GTH1 is follicle stimulating hormone for the mammals, and GTH2 of the fish is equivalent to luteinizing hormones of the mammals over there. So what is the function of the follicle stimulating hormones? Follicle stimulating hormones will induce the small, the germinal cells to become ovum. So that is follicle stimulating hormones. They allow the germinal ones, the spermatogonia or the oocytes to develop into oogonium, right? So that is follicle stimulating hormones. And GTH2 is luteinizing hormones. The luteinizing hormone, what does a luteinizing hormone does in the humans? They help in releasing the egg from the ovary. So same thing over here. They release the eggs from the ovary of the fishes over here, GTH1 and GTH2. So GTH1 is involved in the gametogenesis and steroidogenesis, both male and the females, whereas GTH2 is involved only in the females, that is during final maturation stages of gametogenesis. And these are responsible for the sex steroids hormones, that is androgens, estrogens, and progesterone. Androgens for the male, estrogens and progestins for the female fishes. So that is, they are going to regulate the gametogenesis. What is gametogenesis? Gameto is gametes, genesis is to giving rise. That is called as gametogenesis. Reproduction, phenotype, whether it is a male or a female, and behavioral characters. What is this behavioral characters? A male has a different character, female has a different character. That is the behavioral characters which is taken care of by this gonadotrophin. So that is hypothalamus, pituitary glands, and gonadal axis that we are talking about. So in a simple picture, if you want to understand, hypothalamus induces pituitary gland induces first the thyroid stimulating hormones, or gametogenesis, that is GTH1 and GTH2, or ACTH. What is this ACTH? Adrenocorticotrophin hormone. This is cortisols, which is released during stress. When a person is stressed up, they have this excess of adrenocorticotrophic hormones, that is the cortisol, that is being released during stress or when they are depressed. All these condition occurs over there. Now, this thyroid stimulating hormones, they help in growth and development. Then this pituitary gland also releases GTH1, GTH2. The GTH1 is used for gametogenesis and steroidogenesis, both for male and the female, and GTH2 only for females, that is maturation of gametogenesis over there. Now, if you're going to separate them, hypothalamus, gonadotrophic releasing hormones, pituitary glands, FSH and luteinizing hormones, both affects the ovaries. Ova is produced, and this also produces estrogen, progesterone. Why estrogen and progesterone? When the eggs are released and when the eggs are getting fertilized, estrogen and progesterone are the one that is going to take care of the future development of the embryo over there. So that's estrogen and progesterone. In the male fishes, pituitary glands, gonadotrophin, they release, the receptors are given for the Leydig cells. What is a Leydig cell? If you all have noticed the testis, the TS of the testis, you have this germ cells, and also you have this Leydig cells. What is the function of a Leydig cell? Leydig cell are the one that is going to induce the release of the testosterone, the male character of the fish or humans or anything is a Leydig cells, which will give rise to the testosterone, which gives that male character to them. Once this is released, the spermatogonium is released, and the spermatogonium also has Sertoli cells. Function of a Sertoli cell gives nutrition to the developing sperms. So that is the function of a Sertoli cell. Two important cells in the testis, Leydig cells and Sertoli. Leydig cells are the one that induces testosterone, the male character. Sertoli cells is the one that takes care or nourishes the development of the sperms. That is the importance of Sertoli cell, which gives rise to spermatogonia, and then sperm genesis occurs over there. So if you can see where you find them in the male and the female, if you can see the male over there, so you have the small testes. Can you see the small testes, a pair of testes over there when we take it out? Now this is how a testis looks like over there in a male. And in the female, that is the ovaries, a pair of ovaries. So you can see all these ovaries filled with eggs in them. And that's the reason why they have from thousands to lakhs of eggs can be found in this ovary over there. Okay. So a little bit of a detail of male reproductive organs over there. So you have a pair of testes over there. So this is a pair of testes. Then you have this kidney, the urinary bladder. The testis opens into sperm duct, and then both of them open into rectum and anus over here. So if you can see over there, when you cut open, that's the kidney over there, that's the liver over here, the heart over there, the backbone, and this is the testis, a pair of testes. And below the testis is the stomach, and they open into urinogenital orifice. So this orifice over there is the place where they open up over there. And how do they develop? Now, if you can see over there, this the primary spermatocytes. The primary spermatocytes. Now, if I'm going to take the section of the spermatocyte or the testis over there, so you have this round one over there. Now, this one is a germinal cells. Now, this germinal cells are the one that gives rise to spermatocytes. And once the spermatocytes are being produced, they undergo mitosis. Remember, so that is the mitosis. And once the mitosis gives rise to the cell spermatocytes over there, this spermatocytes undergoes the first meiosis, and that gives rise to secondary spermatocyte. The primary. So this is the primary. The spermatogonia undergoes meiosis 1 to form primary spermatocyte. The primary spermatocytes undergoes meiosis 2 to form the secondary spermatocytes, and the secondary spermatocytes are the one that give rise to spermatids. So what did I say? Primary spermatocytes undergoes meiosis 1 to form secondary spermatocytes. Secondary spermatocytes undergoes meiosis 2 to form spermatids. All right, meiosis 1 and meiosis 2. So under meiosis, two divisions. Mitosis, only one division. Meiosis, 2 divisions. If you have studied the spermatogonia and the gametogenesis over there. Primary spermatocyte, that is spermatogonia, gives rise to primary spermatocytes. The primary spermatocytes undergoes meiosis 1 to give rise to secondary spermatocytes. Secondary spermatocytes undergoes meiosis 2 to form spermatozoa or the spermatids. And the spermatids are nourished by the Sertoli cells. So you have the Sertoli cells over there. They are nourished to form the sperms. So that is the story of testis over there. And coming to the story of the this is you can just go through the reproductive cycle, that is the resting phase, preparatory phase, mature phase, and spermiation phase. This is what I was just talking about over here. And this is how the mature stages of the testis looks like. Now that is the young immature stage where the testis is still developing over there. And then this is the resting phase over here. That is a maturing testis where the spermatocytes are being formed over there, the primary and the secondary spermatocytes being formed. Then that's a mature testis with lots of milk in them. And that is a spent. What do you mean by spent? When they release all the sperms outside, it has become empty. The testis has become empty over there. That is called a spent testis. Something like whenever you have money in your pockets, it is full. But whenever you have money, you just want to spend it away. After you're spending it, your pocket is empty. Same thing over here. The testis is also empty over there. So that is why it is called as spent testis over there. Okay, coming to oogenesis, that is development of the egg over there. So you have this oogonium over there, oogonium giving rise to more amount of oogonia over there. So this is the mitosis phase over there that is going to increase in number. And this oogonium gives rise to primary oocyte. Well, how primary oocyte? That is development. The cell grows in size. And once the cell has grown in size, the primary oocyte undergoes meiosis 1. When it has undergone meiosis 1, it gives rise to secondary oocyte. How you say it's secondary oocyte? Because it is going to give rise to one ovum, large ovum, and one small ovum. And this small ovum is called as polar body. Why small one? Because it has to concentrate only on one egg. Why? Because most of the nutrients has to be given only to one egg. What is the nutrient over there? Albumin and yolk. If you remember when I was talking about the bearers over there, you have this live bearers, oviparous, viviparous, and ovoviviparous, where you have two sets wherein fertilization is internal but nutrition is outside. Incubation, but nutrition was not given by mother. Why? Because these eggs are the one that is having yolk present in them, and this yolk and albumin will take care of the development of the young ones. So for example, if this egg is exactly dividing into two equal cells, that means no two individual zygote is going to grow properly. So the egg, only one ovum, is concentrating excess of albumin and yolk in them, and that is the reason why you are having development of the young ones properly. That is the importance of this one polar body, which is very small. Why? Because they have to divide. 46 has to become 23, and that is the reason why they have to divide. So one will have this 23 chromosomes but less amount of yolk. One egg will have 23 chromosomes but more amount of yolk and albumin. Now this secondary oocyte will undergo next meiosis 2, and again it will give rise to one ovum with concentrated yolk and albumin, and the other one is a polar body with no yolk or albumin but chromosomes are present in them. That is the phases of oogenesis over here. So how is the structure? You can see the ovaries over there. So you have again a pair of ovary. So you have these two ovaries over there, which is opening into an oviduct, and that opens into the anus. So this is when you cut open the fish, you can see these ovaries present in them. And most of the ovary in the teleus, hollow sac-like structures, and the germ cells or the oogonium are present over there, which multiply mitotically. That is what I was talking about in this. They multiply mitotically over there. So that is the mitotic divisions over there, and transformed into non-yolky primary oocytes. And these primary oocytes are slowly covered by two layers of follicle cells, the follicle stimulating layers over there. And they also have this outer thecal layers and inner granulosa layer, which undergoes vitellogenesis. What do you mean by vitellogenesis? I think you must have done this topic even in your practicals. Vitellogenesis. What is vitellogenesis? Can anybody tell me? If you have done this topic, vitellogenesis.
Ability of production of eggs in a particular ovary.
Okay. Anything more? Anything more? Metallogenesis. It's there in the in your slide itself. When the yolk is deposited, that is called as vitellogenesis. So you must have seen in the chicken egg also this yolk. If the fish, the hens are not given enough food, the yolk is very less, and the yolk is almost light yellow in color. But if you give them proper nutrition and diet, the yolk is very much orange in color and more amount of them. So that is what is called as vitellogenesis. Deposition of yolk in the egg is called as vitellogenesis, and the analysis of yolky egg indicates that the egg yolk is composed of lipovitellin and phosphovitin. Lipo is lipids. Phospho is presence of phosphorus present in the. Why phosphorus again? Again, phosphorus is it gives nutrition for the egg for development. So what example is this? You can see this of vitellogenesis that I was talking about. Oogonia nest over there, they're slowly growing in size. So that is multiplication stage that I was talking about, the growing of them. And you have the nucleus present inside. That's a nucleus, and the small dots are the nucleolus. And you have this vitellogenesis, that is oil droplets, lipovitin and phosphovitin being deposited around this eggshell or around it. And those are the theca granulosa and pellucida that is being present around them. So you have this excess amount of yolk. Now, you have this so many nucleolus. Can anybody tell me the importance of nucleolus in the cell? You have a nucleus. Inside the nucleus, you also have nucleolus. But why should this egg have so many nucleolus? Very beautiful question. If somebody can answer me this. You might be asked for any MCQ questions if at all you're going to write any exams in your future for different promotions. Function of nucleolus. Can anybody tell me? You can see in this picture also that is the nucleus, and those small dark dots are the nucleolus inside the nucleus. Girls, come on, you must have done genetics, chromosomes. You must have done this. Are there no boys in my class? Everybody is searching the cell phones for the answer. Nucleolus. Function of a nucleolus, girls. Nobody? Nucleolus is the one that produces mRNA. RNAs, yes, where are the RNAs coming from? RNAs are produced by the nucleolus, right? Nucleus has the chromosomes present in them, but nucleolus gives rise to RNAs, right? So your chromosomes are DNA, but you also need RNA. Why you need RNA? Three types of important RNAs: mRNA, rRNA, tRNAs. All these RNAs are produced by this nucleolus. Why RNAs? Because of protein synthesis. Without RNA, you will not have protein synthesis. So that is the reason why these RNAs are very, very important along with the DNAs over there. And this is produced by this nucleolus present inside. And that is the importance of nucleus and the nucleolus. So you can see over here how they are growing in size with the deposition of the yolk. So you can have this yolk vesicles, the oil droplets that are formed. And these are the different stages, how they develop into the bigger eggs over there. So here you can see when you find these eggs, you don't find eggs in all the seasons of the fishes. So here, when do you find eggs? Mostly from April to June, April, May, June. Whenever you buy fishes from April, May, June, that is right now, you can also buy whichever fish you buy. The females have lots of eggs in those ovaries over there. Why? Because that is a season where you have the oogenesis occurring over here. So example over here, from July, August, secondary growth occurs with the lipid droplets over here. And from November to February, the growth is increasing. That is vitellogenesis is occurring from November, February. And then at March, your granules are there. And from April, May, ovulation occurs, ovulation releasing the eggs. And then you have this young ones. That is the cycle of oogenesis over there. And the ovarian cycle, again, resting phase. Stage 1 to 2 is the resting phase, where they are just multiplying in number. Preparatory phase, where the yolk deposition occurs in the preparatory, and maturation phase, pre-spawning. So these are the zona granulosa and zona radiata, that is covering the egg around them, and then they are released out during spawning phase. So these are the different shapes where you can see over here. So that is the immature or the resting phase, early maturation phase over there, then the advanced maturation, and complete maturations you can find over here. Spawning phase, ready to spawn, that is spawning is releasing of the eggs is called as spawning, and this is the spent phase, where all the eggs are released. So you can see the ovary, a pair of ovary from the covered by the mesovarium. So the ovaries are covered by a mesovarium, thick skin, a very thin layer covering around them. And yeah, you can see the now this is an advanced maturing phase. You can see the size of the eggs of so this is an advanced one, a pair. So this is one ovary over there, this is one ovary over there, a pair of ovary over there present over there. And yeah, you can also go through this reproductive cycle of the ovarian, that is the resting, preparatory, maturation, and spawning phase over there. Vitellogenesis, as I told you all, what do you mean by vitellogenesis? The deposition of the yolk inside the egg is called as vitellogenesis, which is rich in calcium binding phosphoprotein. And of course, it also has yolk protein like lipovitellin, phosphobitin, beta component, and C-terminal peptides. As I told you all, importance of phosphobitin and lipobitin. And that's the reason why whenever we are talking about yolk, we say yolk is having lots of fat present in them because of the presence of lipobitin. But when you're talking about fat, when you're talking about the egg, sometimes those who are wanting to do this dieting, what they do is they boil the egg, remove the yolk conveniently, and just take the egg white. When you're doing so, you are losing a very important lipid that is present only in the yolk, which you don't find in the albumin. So when you want a complete nutrition, you have to have the whole egg as such. Please don't remove the yolk, because you are eating only, it is something like you have removed the juice and you're just eating grass over there. So it is as such. So when you want a complete nutrition, please do take along with the yolk, because yolk also has a complete nutrition present in them, which you will be missing if you're going to throw them out. So that is the importance of yolk over there. And this is what you must have done in the practicals, gonadosomatic index, GSI. That is the weight of the gonads by weight of the fish into 100. So this you must have done. Why do we do this gonadosomatic indexes? Time and again, you want to know that your fishes are hale and healthy, and they are able to give that many number of eggs with in comparison with the weight of the fishes. So this is one more way to check whether you're giving them proper nutrition and whether the fishes are growing in good quantity. So breeding of common carp, as such, you must have seen the difference between katla, rohu. So katla is a big face over there, katla over there, and rohu a little bit smaller face. Katla is a long one over there. This is a little bit narrowed down, and that is Sirina mrigal and Labio calbasu, another type of Labio rohita over there. And distribution of common carp over there. This is the Cyprinus carpio, ranked third among the farming fishes over there, next to only silver carp. And this was introduced by Germany. That's why it's called as European carp. And they grow over 1 kg in 1 year. And as I said, common carps are they can grow anywhere, wherever you want to give them. Now, another important topic is, I'll just wind up very quickly over here, transportation of fish seed and broods over here, how we are going to transport them. Earlier, you must have seen they were being transported by this earthen pots. Why transportation? Because you are in some place, but you need brood fishes. Brood fishes, the ones that are mature fishes, which are ready to reproduce and lay the eggs. Those are called as brood fish. Fish feed is those, either it can be the eggs, either it can be the embryos, either it can be the fries, or it can be fingerlings. What is fingerling? The size of the finger is called the fingerlings. So these have to be transported. So how transportation is occurring? Earlier, they used to transport by this earthen pots, although this is aluminum pots that you can see over here. They used to send them by this, but mortality was more in them. So traditional way, mortality was more, but later on, different types of transportation has come up. So transportation, what causes mortality during transportation? It can be several reasons. What are they? Depletion of the dissolved oxygen, accumulation of carbon dioxide, accumulation of the lactic acid present in them, that is because of the stress, sudden fluctuation in the temperature, osmotic imbalance, or diseases. Why? Crowdness, disease can occur. So all these can be a cause of mortality. So to reduce that mortality, what can be done? So you can just go through all these things. If at all you want any questions to be answered, you can ask me in the next class also. Now, lactic acidosis. This is one important point I want to talk about. When we are transporting fishes, because of the excess of fishes present, they have this excitation, stress, and this leads to lactic acid release in the water. When the lactic acid is released, the water content becomes acidic, and this acidic content also in the blood increases, and that leads to collapse of the fishes. So lactic acid is one more important point that you'll have to understand when you're transporting the fishes over there. So you have to do the conditioning. So how is the conditioning done? Conditioning is done by, when you're taking any packet or any earthen pots, you release the fishes slowly one by one in them, leave them for 24 to 48 hours so that they get used to that environment where they are being set up. Take care of the oxygen, take care of carbon dioxide production. Don't feed the fishes for almost 24 to 48 hours. Why? If you're not going to feed them, they're not going to do the digestion. If you're not going to do digestion, excretion is also less. So conditioning. So these are some of the conditioning that has to be done before transportation of the fishes. Method: two types: open system, closed system. Earlier, this was the open system that were being done, and this open system had its own disadvantages also, because of excess of movement in them. They can die, less oxygen. All these can occur in the open system. Now later on, so this is how the open system occurred over there. In the closed system, this is the best part of transportation in the closed system. Why? You're going to use polythene bags of various dimensions. So you can see over there, so polythene bags can be used. In the polythene bags, what you're going to do is you're going to fill 1/3 of the polythene bag with water. 2/3 of them is going to be oxygen. You're going to pump oxygen into these packets. And what you're going to do is you're going to twist the packet, bend it up, and put a rubber band. Now you can see that you have on both the sides this type. You'll be wondering, polythene covers one side, it is always closed one. It's only the other side which is open. Then why do this type over there? Why? Because the polythene bag, when it is present in open space, the edges have very small space. Some of the fishes can go and get stuck up inside them, and they cannot come out, leading to the death. And that death will again increase the diseases in them. So that is the reason why when you tie it up below, you have an uniform shape over there, and there are no death in those fishes over there. Now this polythene bag is kept in boxes like this with insulation, and time and again you're going to check for the oxygen levels in them, and these are carried to different parts. So these are the packets where you carry them to different parts over there, and they are sent to different places. So these are the different types of containers over here. So this is a 25-liter container. This is a 50 to 150-liter container. You can see those places for oxygen supply. You have also pipe for oxygen supply over there. And this is the open type over there. And this can be distributed either by flight or by transportation through trucks over there. So releasing part and all, I'll talk about how you have to release them in the water in the next class over there. So these are some of the ways of transportation of the fishes over there. So with that, we come to the end of the first topic, that is block 1. We have completed. Any doubt, girls? I hope you must have understood a little bit of what we were talking about. Yes, students, any doubt? So if there is no doubt, then I'll take it as you have understood the class as such till now. And in case you still have any doubts, you can just get back to me in the next class, or also I think you have my number. You can just message for the different questions if you have. Yes, girls. So meet you all tomorrow for the next topic. Good evening. Okay, girls. Shall we leave the class now? Yeah, thank you. Thank you for, if it is informative, yes, I'm very happy that you have understood the class as of this day. Many more to come with a lot of information. Make sure that all of you all join all these 7 days, because this is going to be a really informative class, and it will be easy for you all to write your theory exam also if you all are going to sit and listen to my class. And if you all want this PPT, I'll be sharing that, provided you give me the group, which group you all are in. I will share my PPT into that. Okay, girls. See you in the next class. Bye, girls.
Thank you, ma'am.
Thank you.